Weathering Change seminar series. Our speaker today is Dean Jones from Emory University. His talk is The Human Exposome and Future Health. I'll turn it over to him now.
Okay, thank you, Kermit. Yes, so I have too many slides as usual. I wish I were there in person because I enjoy the direct interactions, but this is more efficient with regard to being able to reach more people. Let's see if I can. Oh, my slides are not advancing. Okay, there. I have no conflicts of interest, financial or otherwise. I have many acknowledgments, and none of this would be possible without the support, mostly from the federal government. And I have to acknowledge the phenomenal scientific team that has contributed to this work and is contributing to the work. So there's three main points. I'm going to briefly review the history of the exposome, and then I'm going to talk about the needs and strategies for exposomics and other technologies to really advance our understandings of mechanisms and potential health benefits. And then I'm going to finish up by just sort of a brief perspective on where we are and how I think we have to move forward to utilize the information from the exposures to improve health. So with regard to a brief history, I wanted to start with this slide. This is some of you will recognize that guy standing in the middle there, Dan Niebert. He was one of the students of Professor Howard Mason, who was also my PhD mentor. And my story on this really begins with my training. Howard was a phenomenal scientist, and he made this statement: "You have the greatest opportunity to contribute to knowledge by identifying critical unanswered questions for which methods are not available to answer. Then, if you develop the methods, you contribute by providing the method and also by answering the critical questions." So Howard introduced the term xenobiotic, and what is important is that when Ron Estabrook reviewed this back in 1982, just a little while ago, he recognized that there were 60,000 chemicals used in commerce. And so the key question is, if there are 60,000 chemicals that could potentially be important for human health, how can 60,000 chemicals be measured? And so when I got into this specific research area, ultimately leading to the exposome research, it was in 2003. And at that time, the best analytical laboratories in the world could measure only in the range of 200 to 300 metabolites or chemicals. And those were done with multiple analytic methods, and would take weeks to get that number of measurements. And so that what was remarkable was that in 1957, Morenstein introduced the automated amino acid analysis, and that was going from one chemical at a time to measuring 30. And that was such a dramatic improvement that they ultimately received the Nobel Prize, not exactly for that, but it was directly a consequence of that development of the amino acid analysis. So important thing is that in half of a century of progress, the world had only gone from 30 to 300 metabolites in capability, and these were being measured one chemical at a time. And so if you start thinking about trying to measure 60,000 chemicals, the time and the expense would just be considerable. And so I had the good fortune at that time to go to NIH to listen to Elias Zerhouni, who was the director at that time, introducing the NIH Roadmap Initiative. And he gave this just a brilliant talk on what he called the data of the future. And he basically said that for the future, we needed to have simple assays that were high throughput and automated, that gave extensive coverage, were highly reproducible, were standardizable for cumulative libraries, and were affordable. And so in 2004, I decided to adopt Zerhouni's concept and really try to move forward with omics scale metabolomics development. So to move beyond that 300 limit to go to thousands. And so I put in a grant proposal on this to NIH using the ultra high resolution mass spectrometry as the approach to do it, and got this brilliant uh, review that this is the type of thing that we live for, or is our peer review from NIH grant proposals, a waste of a good instrument. And so obviously, that was a setback. The next year, however, there was this very, very exciting editorial by Christopher Wilde on defining the exposome as the complement to the genome to be able to account for chronic disease. And what I didn't realize was that that concept of what I wanted to do on metabolomics and Christopher Wilde's exposome were really on a collision course. And basically, what happened was I went forward with the concept of developing this high resolution metabolomics capability. And Gary Miller was putting in a program project grant on environmental contributions to Parkinson's disease. And he needed a human clinical or human component to that project, to the program project. And so he asked me if I would put in a project on measuring the uh, metabolic consequences or metabolic changes associated with environmental Parkinson's disease. And so what I put in was this ambitious proposal that we were in 5 years. It had taken 50 years to get a tenfold increase. So I considered it very ambitious to try to go for a fivefold increase in 5 years. But really, the more ambitious component of this was that I decided that we had to be able to do it in 10 minutes, in a 10-minute analysis, not weeks, but 10 minutes. And so we got the award, and through the collaboration with Tianwei Yu, he developed software that would enhance the ability to use the mass spectrometry data. And with Jennifer Johnson, a PhD student at the time, using the methods for analysis, what we found was that indeed we had in 1 year achieved the fivefold goal. And over that 5 year period, with the help of Quinlan Salto and Karan Oakbal, we were able to take that to 100-fold improvement. And so this was really a breakthrough in terms of the analytic capabilities. But what it showed was that those analyses of human samples were measuring far more than metabolism. They were measuring non-nutritive chemicals that were found in the diet, so dietary components. They were measuring microbiome-derived metabolites. They were measuring metabolites from dietary supplements, from drugs, therapeutic drugs, and the drug metabolites. They were measuring, the methods were giving us commercial products, environment, or chemicals, as well as things from environmental products that we were consuming or being exposed to, such as the one that really hit me was when we found that we could detect who was a pet owner and who wasn't a pet owner by the levels of the insecticides that were in the flea and tick collars at that time. And then we could get a whole variety of different environmental chemicals. So this was in I put this figure in a review in 2012. So already at that time, we had already merged the concept of the high resolution metabolomics with exposomics. We didn't have a clear picture of it at that time, but obviously it was there. And this was the what I call the seminal paper, was a breakthrough on conceptually. And this was a project led by Youngja Park, who was at that time independent, well, not independent, she was a collaborative scientist within the group, within the clinical biomarkers laboratory. But she had access to plasma samples from 7 different mammalian species, 3 barnyard animals, 3 laboratory animals, and humans. And so the concept was that we believed that the humans were exposed to such greater diversity of chemicals than other animals, the laboratory animals or the barnyard animals, that what we would see 5 times as many chemicals in human blood than we saw in the blood of these other 6 mammalian species. What the data showed was really rather remarkable, and that is that no, that's not the correct answer. There's almost exactly the same number of chemicals detected in these 6 other species as in humans. And what it showed was that only a small fraction of those were actually intermediary metabolites, and those were relatively common among the 7 species. The remainder were environmental chemicals. And what the analysis showed, the bioinformatics and biostatistics analyses on these 7 species, when we compared them, what it showed was that there was a correlation or an association of the environmental chemicals that we could measure with the biological responses. So those metabolites that we were seeing were actually correlating in patterns with the chemicals. And so what this led to was really the development of the exposome research paradigm. And that is that we have external exposures, and many of those result in an internal dose of that related to that exposure. And then those internal doses are causing metabolic responses. And that's the paradigm then, is those metabolic responses are what are responsible for disease and disease risk and health outcomes. So this is the paradigm that we work with. And this was what we had already developed in 2012. And so in collaboration with Gary Miller at that time, we recognized that Christopher Wild's definition of the exposome did not include the biological responses. So we refined the definition at that point that the exposome is the cumulative measure of exposures and biological responses. And what we recognized at that time was that we really had a problem. And that is that we could measure 50,000 mass spectral signals, but essentially all of those signals were unidentified. And this created a tremendous roadblock as to how to move forward. We had the analytic capabilities, but the task of identification was simply overwhelming. And Shazal Li was at that time was sorry. Shazal Li was a postdoc at that time with Bali Plundran, And he was really very interested in the metabolomics, and he worked with us to develop this program, a pathway enrichment tool for using the untargeted mass spectral data to map, to use permutation testing to map to network and metabolic pathways. And so this was really transformative because what it allowed was us a way that we could extract information from this massive amount of mass spectral data and could then move forward with the understanding of what chemicals were associated with what metabolic responses. And so that concept was extended through work that Young Mi Go was really pioneered. She had developed methods for measuring redox proteomics in the group, and so she was really an expert in measuring the way that proteins respond to oxidative exposures. And so she wanted to relate that to the metabolic changes. And so this was the concept, was that if we have different omics layers, can we integrate those? And Karan Uppal was a PhD student at Georgia Tech at the time, and he was working with Youngja and us trying to develop improved methods for the high resolution analyses. And what he developed, really a prototype in 2013, was this prototype of what is now available as XMWAS, which is a multi-step procedure where you can take different omics layers and integrate them and use them with community detection tools to identify which metabolic pathways and which biologic functions are varying in association with a specific or multiple environmental exposures. And so this tool was incredibly powerful. Um, and what Young Mi did was she went on to verify it, to actually test it in model systems. Does this tool really work? And what she did was she used it to integrate redox proteomics and metabolomics data from mice that had been treated with cadmium. And what she found was that there was a central hub of proteins or peptides from enzymes and transporters of fatty acid metabolism. So these were very clearly defined by mass spectrometry-based proteomics, and so these were very rigorously defined, and they correlated with a very, very tight cluster in response to the cadmium exposure. Now, what was really exciting was that the blue rectangles here represent different metabolites that are correlated with those peptides, with those proteins. And these are enriched in acyl-CoAs and acyl-carnitines, which are, of course, the transport components and the metabolites that are involved in fatty acid metabolism. And so it was really saying that from the cadmium exposure, the central response structure was oxidation of mitochondrial proteins involved in fatty acid metabolism and associated changes in those metabolites, indicating that we were really getting inhibition of these enzymes and transporters, and it was being directly reflected in the levels of the acylcarnitines and acyl-CoAs. And so what she did was she used stable isotopic tracer methodology. That's the beauty of mass spectrometry, is you can not only directly measure chemicals or metabolites, but you can also trace their interconversions. And so she was able to use stable isotopic tracer methods to, in fact, validate that this integrative omic structure is indeed reflective or measuring what we think it's measuring, and that is cadmium effect on the proteins and the consequent changes in the metabolism. So we have, over the past several years, I'm not going to talk more about this, but just just give the central concept, we have extended this extensively to look at the relationship of transcription, the proteome, and metabolome. And in fact, the data show that this is a fully integrated network response. And so if you have something that is perturbing at any level, it is causing perturbations at all of the levels. So in fact, it is responding as an integrated network. And we'll come back to that in a bit. So the main point in the history, at this point, we already had integrative omics that provided a validated approach to understand the relationship between environmental exposures and metabolic health. Before I finish up on this introduction of the history, I have to refer to work that Doug Walker did when he was a PhD student working with us. He was a PhD student at Tufts, but in engineering, but he did most of his thesis work with us, studying and developing these methods for exposome research. And he introduced gas chromatography. Everything that I've talked about up to this point has been liquid chromatography based measurements. He introduced the gas chromatography measurements and produced this slide where he claimed that we were approaching 1 million in terms of the coverage, that we could actually get 1 million signals from a single biological sample. And so this really just catapulted everything to an incredible new level as far as the analytic methods. The other critical development that was also in this, the first decade of this exposome history, was the development of the quantification methods. And Youngmi Go also introduced methods for being able to quantify environmental chemicals. And what that data showed was that it was possible to quantify environmental chemicals over 7 orders of magnitude in human plasma or human serum. So it was really quite dramatic in terms of being able to just move the entire field for exposome research forward. And so most of that was summarized, or a lot of that was summarized in this review article from 2016, which sort of captured the state of the art at that point. So summarizing, brief history, exposome methods were extensively developed and validated already in 2016. Most critical point is that human exposome includes far more than just the low abundance environmental chemicals. And what we have to look forward to is development of new methods that are going to address the complexity of the cumulative exposures and also integration of those environmental exposures, those environmental chemicals, with variations in diet, therapeutic drugs, health behaviors, and all of the other exposures that we have. So I'm going to spend a few minutes now on exposome epidemiology and the really how we're moving forward currently, what's been done in the past 10 years in this. And the goal is to make what we're doing today obsolete, because we need new methods to deal with those issues of the cumulative exposures, cumulative lifelong exposures, and all of those integrative interactions. So one of our major challenges is humans live a long time. The best approach that we have today is to use archival samples. And so we've done a lot of work in collaboration with Barbara Cohn, who's the director of the Child Health and Development Studies. And she has these samples that were collected in California between 1959 and 1967. And so if we have outcome data for that cohort, then we can really understand long-term consequences of whatever exposures there were in 1959 to 1967. Now, that's obviously a limitation in that the exposures between 1967 and today have changed considerably. But on the other hand, it does give us a way to understand and developed the methods to be able to understand the exposome and long-term consequences. So her research has already shown that in the maternal life that there's an association of DDT exposure during pregnancy with subsequent breast cancer outcome. She's also shown, her group's also shown that in the F1 generation in the daughters, there's also an increased risk of the in utero exposure to DDT to subsequent breast cancer. And she's also, her group's also shown that in the granddaughters, that there is earlier menarche and, or there was earlier menarche, and that they had uh, increased BMI associated with the DDT. So clearly very important, and that these cohort studies with archival samples and outcome data, I think are what's going to really be useful to fill the gaps as far as understanding of the exposome. So the second challenge is that there's many types of exposures, and I already indicated 60,000 commercial chemicals already in 1982. So we have to deal with that there's so many types of exposures. And so we've tried to approach it by developing capabilities for measurement. And Ken Liu developed methods that we extended to actually characterize almost 900,000 metabolic products. Most of these we haven't detected in human samples, but we do have the reference information. We've developed capabilities. Will Crandall just defended his PhD last week, but he had this very nice paper that came out recently showing how to use biotransformation to enhance understanding of natural products. This is a major difficulty when you have a natural product of food or other product that has thousands of chemicals in it, and then when we are exposed to that, we biotransform those thousands into tens of thousands. And so he developed a multi-layer molecular networking approach to be able to understand and identify those metabolic products. Again, a vast number. We published last year, Jen Gion published a paper on over 1,000 FDA-approved drugs that we had profiled for their metabolites as part of an NIH grant a few years ago. And she provided this database, which contains, I believe, in the range of 70,000 drug metabolites. So it's possible to actually go in and measure drug metabolites for over 1,000 drugs in human samples. So we now have capabilities to really understand the distribution of drugs in these archival samples. And then Shen Hu has further developed those methods that that Doug Walker had introduced on using gas chromatography, and has now shown capabilities to measure thousands of environmental chemicals with gas chromatography methods. So the challenge of exposures and co-exposures, well, we're moving forward with capabilities, but there's still a long way to go. And I'm just going to give you a little bit of a touch on how we're doing that. This is from going back to that study, the work with Barbara Cohn's archival samples from the child health development studies. We're looking specifically in the next few slides at the maternal breast cancer outcome. And this was work that Young-Mi Go led on trying to understand the exposure metabolite communities associated with breast cancer outcome. And what she did was she selected those environmental chemicals or commercial chemicals that were positively associated with subsequent breast cancer diagnosis. And then she used XMWAS to identify which which other metabolites are associated and which metabolic pathways are associated. And so one of the communities of interactions that she found was the one shown here, community one, which included an interaction of an insecticide with a commercial product and a food-derived metabolite, as well as an endogenous metabolite with these pathways associated with amino acid metabolism. And this whole network structure was what was associated with the breast cancer outcome. If you looked at each individual component, there was nothing that survived an FDR threshold. So there was nothing that would pass false discovery rate filtering for a statistically significant association. But the overall community structure was significantly associated with outcome. Another of two more of these communities, one was associated with retinoate metabolism, and another was associated with some unidentified chemical. And these communities, instead of being associated with amino acids, they were being associated with lipid metabolism and signaling lipids. And then finally, there were two other communities. One of these communities, which was the one that just terrified me, was this fungal product, and the community 4. It was a very strong community of metabolic associations. And what was most remarkable about it was that it mapped to ergosterol or vitamin D2. And we could not get definitive identification. So it appears to be related to vitamin D2. But the thing that really terrified me about this is that in principle, it's showing that ergosterol, vitamin D2, or some closely related metabolite was actually linked to increased risk of breast cancer. We don't know that that's true, but that's what the data suggests. And of course, vitamin D2 is still sold, and this would be a very small effect size from these types of analyses. And so don't go crazy as far as thinking about this, but just be aware that this is the type of information that we're able to now extract from looking at archival samples with health outcomes. The other one on here, which is equally terrorizing, is the glucose association, and a common food preservative that was used at that time in this community 5. And so what it's telling us is that there are low, small effect size associations that we're seeing that we don't know whether that small effect size is because it's highly important in some individuals or whether it's just a small risk in all individuals. But these are the types of things that we're now able to pull out from the exposome research. Of course, what one has to do is take those type of correlative data. Associations do not establish causation, so we need to come back and look at that in a targeted manner and to test the hypothesis. So I'm going to spend the last few minutes thinking to the future. We have many exposures to which we have no way to avoid the exposure. These are things like air pollution. It's here. We have done a lot to be able to clean the air over the past half century, but the reality is we still suffer from air pollution. There still are wildfires. There still are lots of conditions where we just don't have control over what we're exposed to. And so what we've been thinking about is how do we, if we can't eliminate the source of the exposure, can we prevent or remediate the adverse biological responses to those exposures? And the question is, can we use exposomics approaches to guide health restoration? So that's the thing that we're thinking about now, and I'm going to just give you some of the ways that we're approaching this. So the concept is that during evolution of multicellular organisms, environmental exposures were an important driving force. And so we have to accept that if it's an important driving force, there's going to be biological responses of all types in all cell types. So it's going to be something, it is going to be a lifelong issue that we have to address, and from an exposome standpoint that we need to understand. So yeah, this just illustrates that same point. So put in my bias. My bias, of course, I may not be around for the next, 45 years to continue developing new methods, but my bias is that we must prioritize development of affordable methods for global exposome assessment. And we need to do this as quickly as we can, and we need to get this into our annual health checkups, because this is what we're going to have to be able to understand what our individual exposures are, what our individual risks are, and eventually how we can go about intervening to protect against those risks. So I'm going to illustrate it by talking briefly about the global increase in early onset cancer, the whole bunch of them, where over the past 30 years, we've had 64% rise in colorectal cancer. Just you can go down the list. I'm going to specifically talk about thyroid cancer in a minute. Almost 80% increase over the past 30 years. So quite incredible. And so one of the studies that we have ongoing, this is Mary Nellis and Sammy Tini are doing this work. It's again using samples in collaboration with Barbara Cohn at the Child Health Development Studies. These were samples that were collected, obviously back 1958, 1967, and they are samples that were from men who were fathers in the study, and who were then, through the California Cancer Registry, were found to have had prostate cancer, either with or without prostate cancer for the study. And we used the high resolution exposomics to measure environmental chemicals in those archival samples. We used Sammy Tini's new tool, Peak Walk, which is the manuscripts in preparation on that, which is a tool to support high throughput exposomics. And what it has a unique capability to detect chemicals with low stringency and also with high stringency. So you get an idea not only of how confident you are in that chemical and its measurement, but you also don't throw away chemicals because you're not quite as confident about their identification. And then using basically widely available XWAS biostatistics and bioinformatic methods. And what those data show is that the average person, the average man in the study, had in the range of 200 environmental chemicals that were detectable in their serum samples. And there were over 300 chemicals totally detected, and there were in the range of 100 chemicals that were detected most of the time. So it means that there was extensive, widespread, and diverse environmental chemical exposures in this population. When we map this out in terms of the quality of the of the analysis in terms of the mass spectrometry identification and then the frequency of detection, the high quality detection, what we can see is these were all filtered by statistical significance before they were plotted. So all of these chemicals were significantly associated with prostate cancer, and some of them with much greater concern because of their frequency of detection and the quality, the certainty that we had for the identification. And so what this is telling us is that the methods are good enough that we could really begin to map things out, prioritize things in an untargeted manner, because we can now measure hundreds of environmental chemicals using these tools. Jumping forward to today, we have a ongoing study that Josh Preston is leading with Ryan Smith on thyroid cancer, where we're measuring the body burden. Specifically, we're measuring the environmental chemicals in the tumors themselves. And these are measures of 5 different chemicals that are known carcinogens, group 1 carcinogens by IARC classification. And you can see that for all of them that they're higher in the tumor samples than they are in cadaverous thyroids. Although we have to say these are a very small number of the cadaverous thyroids, a non-tumor tissue at this point. We just don't have we have not collected enough samples to really know those numbers. But the reason I'm showing this is really this slide on the right here. The papillary thyroid cancers are the ones that have increased so dramatically. And what these data show is that I believe there's 12 different possible and known carcinogens that we are measuring in those papillary tumors. And what you can see here on the left is that there is a vast array of different environmental chemicals that are measurable in those tumors. And so what it's really telling us that we have the methods and we need to apply them to really understand the body burdens that people have with these different chemicals. So if we can now measure the body burden, how do we address the problem? And so that's the last couple of minutes I want to spend on this. Basically, the concept is that we have adverse exposures, and those cause biological responses that are linked to outcome. What we know from that biological responses of the transcriptome, proteome, metabolome is that it's all an integrative network. And so the question is, can we identify rectifying exposures to protect against those adverse exposures? So that's my belief, is that this is doable and that we must begin to try. So I'm going to use one example, and that is based upon work that this is ongoing. This is one of those, why am I still working? It's because of my passion lies here. A few years ago, we found that the human microbiomes work was initially done in mice, but we know that this is also true for humans, that the microbiome, intestinal microbiome, produces a chemical called valerobetaine from dietary lysine and trimethyllysine. And so what we found in this paper, we showed in this paper, is that this chemical causes obesity in mice and is associated with obesity in humans. And what was most important about this, I think, is that if we take that valerobetaine and add it to mitochondria in a seahorse assay and use a fatty acid to support respiration, what we find is that the valerobetaine is a potent inhibitor of mitochondrial function. And the value here, the range is between 10 and 50 micromolar in these cells. And what's important is that we have measured up to 37 micromolar in human plasma. So that means that some people have concentrations where it's almost a certainty that the valerobetane is inhibiting mitochondrial function. So we're continuing on to this work. Jacqueline Weinberg, who's a PhD student in the lab now, just getting ready to defend her dissertation, but she has made this very important finding that the valerobetane is converted to an analog of carnitine, which we've named homocarnitine. And what she finds is that when the valerobetane is converted to homocarnitine, it causes dramatic changes in short chain acyl carnitines. So in other words, these acetyl carnitine, propionyl carnitine, and so forth are very important in mitochondrial function and other functions in the cell. And with exposure to the homocarnitine or with the valerobetane conversion to homocarnitine, there's a dramatic change in the concentrations. So it's clearly impacting carnitine metabolism and carnitine-related fatty acid metabolism. And the other important aspect of this is that work by Josie Lim, who's another PhD student in the group. She's found that certain foods, specifically from food frequency studies, foods of the Western diet are in fact associated with the high plasma of valerobetane. And so this is saying that it's a reality in terms of associations of food and valerobetane and disruption of carnitine metabolism and mitochondrial function. And I think the take home from this is that in the 1960s, hybrid corn was introduced, and the hybrid corn was just a miracle in terms of agriculture because the hybrid corn with high lysine content provided much improved nutrition for all of our animal products, all the beef, the pig, pork, poultry, just across the board. It provided better nutrition for the livestock. It caused lower cost for food production, and it gave better food products. And so this just revolutionized our agriculture and our food supply. But what we're considering as a possibility is that one of the consequences of that hybrid corn with the high lysine content is that it increased microbiome production of valerobetane. And with that increased microbiome production of valerobetane, it caused a generalized inhibition of mitochondria. And we've found mitochondria, different cell types, are inhibited by the valerobetane. And so if this impairs the metabolic resilience of the cells due to the impaired function of mitochondria, we effectively have a Warburg effect, where we're causing a stimulation of aerobic glycolysis due to that impaired mitochondrial function. And so what we don't know, but at this point seems a real possibility, is that this has contributed to a widespread change in age-related disease and multimorbidity in the last half century. We've linked it to obesity, and it could be contributing to the epidemic in obesity, the associated epidemic in type 2 diabetes. It could also be, and we have data on this, that it causes cognitive impairment in mice associated with cognitive function in humans. And the question is, if it's a Warburg effect, is it contributing to those early onset cancers? So anyway, this is where we are in the thinking. We don't know. We don't have an answer. That's why there's a big question mark there. But what's important as far as the take-home message is that if this is a problem, we actually have a potential solution because we can compete by providing more carnitine. Because if homocarnitine increase is decreasing carnitine, then potentially provision of carnitine would reverse that problem. So this wonderful product called Alcar is available, and there's a fantastic list of claims for its function. Um, the question is, does it work to protect against vilarobetane? And so we've started experiments in terms for mice looking at this, and this is also work that led by Josie Lim in the group that I already acknowledged before. But it's a team effort. And what you can see from this is that if you add vilarobetane, you get this dramatic change in the carnitines, acylcarnitines in mice. And this impacts really short chain, medium chain, and long chain acylcarnitines. And now it is not moving forward. Let's see if I can get this. It's not happy. All right, there. So now if you add Alcar, you provide Alcar to the mice, what you see is that you reverse those effects. And so obviously very early on, none of this is published, it's very early on in terms of discovery, but it's saying that potentially we could reverse the adverse effects of the exposures. And so that's where we're headed, exposome medicine, the use of exposome measures to aid in the diagnosis, management, prevention, and cure of disease. I think this is a very sound way to think about how we can approach some of these environmental exposures, the increasing environmental exposures that we're having, not only with climate change, but with many other aspects of human existence on the planet. And so I will summarize by saying we have a substantial foundation to incorporate the broad range of lifelong exposures into the management of human health. Exposome epidemiology has advanced with integrative omics and network medicine, so we can really be able to address the impacts of cumulative lifelong exposures. And for the future, what we need is new strategies to address the complexity of the exposures and especially remediate the adverse effects and deliver exposome medicine. Lots of people involved here, and I will leave you with just a pretty picture. And what I usually start with, but I felt I didn't have time today, but don't be limited. Don't let your vision limit what you can see, because we have so many unknowns that we have to address. So I will stop there, and if there's any questions, happy to try to answer them. And yes, so
George,
yes, thanks, George.
go ahead. Thank you for the presentation, by the way.
Oh, thank
You're
you.
muted, George. George, you're muted.
That was wonderful, Dean.
Thank you.
One of the things I wanted to ask you about was that mitochondria use fatty acid as a backup mechanism of metabolism, not the primary. And so when you did your experiments with fatty acids, you showed that the mitochondrial function was inhibited by VB, whatever that is exactly.
Vilarobetane, yeah.
Yeah, I was wondering if the normal normal glucose metabolism of mitochondria was affected. It was one question I had. And the second question is, do you think that this is important for when you have a stress condition where you need to use fatty acid metabolism? It's kind of a backup system to energetics.
Well, I will say that it's ongoing research. We're very excited about it, but obviously there are limits. And what I think the biggest issue is that those short chain acylcarnitines do a whole lot more than just support energy metabolism. Um, the acetylcholine biosynthesis, for instance, is drastically dependent upon the acetylcarnitine pool, because that's basically providing the buffer that maintains the acetyl group for the generation of acetylcholine. So things like that are things that we just don't know how important they are because we haven't studied it. But clearly, it's going to differ depending upon the cell type and differ depending upon the needs for those other acyl groups. So yes, early on, we don't I generalized on this, and I did that just to point out that it's exciting. But yeah, we have to approach it with caution, and we're going to have to work out lots and lots and lots of details. So that was the first part of your question. Second question part was that was mitochondrial function
Under stress. When
under stress.
Right, fatty
Yes.
acid metabolism is more of a backup system compared to the normal mechanism. And so if it's altered during periods when stress occurs, that would be disease oriented
Right.
effect.
Yes, that's definitely true, and we do see that. For instance, with cadmium, if you put cadmium plus valerobetane, cadmium also inhibits mitochondrial function, and you're far worse off when you stress the system that way. So definitely that is important in the multi exposures. So yeah, there's lots to do. You need to get back in the lab, George.
Yeah, it's wonderful to see you, Dean, and that's a beautiful presentation. I think you bring a lot of great science to this field, and we're really appreciative.
Okay, thank you.
Gloria, did you have a question? I know you had your hand up first.
No, I didn't.
Okay, John?
Yeah, sorry, hold on. Let me get my camera on. Yeah, this is a great presentation. I have way too many questions to ask, but it would be awesome to play around more in this area. But just kind of, I guess maybe trying to kind of wrap my head around some of what could be done. I'm thinking specifically about the current issue of colon cancer in young adults and the kind of shocking rise in incidence and prevalence of the disease. Have you guys done any work looking at the environmental fingerprints, I guess, for people with colon cancer in terms of the metabolomics of, say, chemicals in the plasma or whatever? And then kind of related to that, I guess the question that one question that I'm really curious about is for these metabolomic fingerprints, how consistent are they from individual to individual across races and different demographics? Is there a lot of variability, or is it pretty specific? Like you can look at one of these fingerprints and say, well, that's definitely this chemical. And how many chemicals have we done this for?
Well, so this is all really new, but we're getting pretty consistent data for in the range of 100 to 200 chemicals that we're seeing, widespread. The challenge is that the exposures have varied over the past 60 years. And so when we do cohort studies, the samples that were collected in the early 2000s are very different from the samples that were collected in the 1960s. And so that's one of the aspects that we're dealing with, is that there are changes over time. And Ann Haynes has a lot of data on that showing the changes over time. But as far as the individual analyses, for individual samples where we have gone in to validate, we have really good data that you have convincing data that these chemicals really are there. and you can get pretty good estimates of their concentrations. So I don't think we have a problem on that. I think the problem is simply um, there's so many exposures and there's so many outcomes and there's so many variables in there that we're just going to have to accumulate lots and lots more data to be able to give you good answers. But it's good enough now that if you have suspect chemicals, that you can come back in with targeted analyses to confirm those. And so I think that's the idea is we're moving forward with on a discovery basis with the expectation that more targeted analyses will have to be pursued. Does that sort of answer your question?
Yeah, and I guess the other question that I brought up was just, have you guys thought about this in the context of the changing demographics of colon cancer?
Okay, so we've done a little bit of work on that now. It's a collaboration with one of Barbara Cohen's former trainees. I'm blanking on her name right this second. But anyway, what she found was that there is an association with certain of the antibiotic use that was used way back then, not being currently used. As far as the environmental chemicals, we did put in a grant proposal on that, but it was not funded. So
Okay.
we don't have that data, but that's doable. It's doable not only in terms of archival samples with known outcomes, but it's also doable with regard to colon biopsies and colon tumors samples. So we could, for instance, today, if we had a surgical team that was interested in collecting those samples, yes, it would be, however many colon cancers they deal with, it would take, that period of time to be able to get the analyses of what chemicals are in the cancers, what chemicals are in the adjacent normal tissue. And, it could do that also with, I'm sure there would be enough volunteers to just give colon biopsies so that one could get a general description of what the variations are.
Mm-hmm. Cool, thanks.
Yeah. I will say just one thing. I just realized we did do some liquid chromatography, not GC chromatography, on some colon biopsies. And the thing that was that struck me more than anything else was that aspirin and salicylic acid was in almost every sample. So that was an interesting one. All right.
Any other questions? Well, thank you, Dean. This was been really good. I really appreciate it. I actually learned a lot from it, being outside my area. So I really appreciate the level that you were able to get it to. So thank you.
Okay. Thank you. Pleasure. Take care.
Bye.